Welcome to the stream of knowledge. This is part 2 on culture media lectures. In our previous lecture we have learned what is culture media, why culture media is used, characteristics of culture media. Today we will learn types of culture media. What are the types of culture media? You should remember this. Based on their consistency or physical state, there are 3 types of culture media. A. Solid medium. B. Liquid medium. C. Semi-solid medium. Based on chemical composition, there are 4 types of culture media. A. Simple medium. B. Complex medium. C. Synthetic or defined medium. D. Special media. Special media has further 6 types. 1. Enriched media. 2. Enrichment media. 3. Selective media. 4. Indicator media. 5. Differential media. 6. Transport media. Let's discuss types of culture media in detail. Culture media is on the basis on the physical state. Number 1. Liquid medium. In liquid medium there is absence of agar, or you can say a medium without agar. It is used for the proliferation of bacteria. For example, nutrient broth. Number 2. Solid medium. In solid medium there is 1.5 to 2.5% agar. It is used for the isolation and identification of bacteria. Example of solid medium are slant, petri dishes, plates, nutrient agar, blood agar. Number 3 is semisolid medium. Semi-solid medium contain 0.3 to 0.5% agar. It is used for the observation of bacterial motility and preservation of bacteria. Example of semi-solid medium is SIM or sulfide indole motility. We have learned culture media types on the basis of physical state. Now we are going to learn types of culture media on the basis of chemical composition. Number 1. Simple media or basal media. For example, nutrient broth and nutrient agar. Nutrient broth consists of peptone, yeast extract, NaCl. Now the question is, what is difference between nutrient agar and nutrient broth? If you add 2% agar in nutrient broth, this will be nutrient agar. As given below, Nb plus 2% agar equals nutrient agar. Number 2 is complex media. Complex media have added ingredients. It provides special nutrients. Number 3 is synthetic or defined media. Synthetic or defined media prepared from pure chemical substances, and its exact composition is known. For example, peptone water. Peptone water is made up of 1% peptone and 0.5% NaCl in water. Number 5 is special media, and special media has further 6 types. We are going to discuss all of these, one by one. First type of a special media is enriched media. In enriched media, substances like blood, serum, egg are added to the basal medium. It is used to grow bacteria that are exacting in their nutritional needs. Example of enriched media is blood agar, chocolate agar. Now the question is, what is blood agar? Blood agar is nutrient agar with 5% sheep blood. It is used for the cultivation of fastidious and non-fastidious bacteria. It is also a differential media. You can identify bacteria through hemolysis of blood and blood agar. Some bacteria secrete enzymes that lyse red blood cells called hemolysins, and this hemolysis is appear as a clearing around the colony. There are 3 types of hemolysis. 1. Beta hemolysis, which is complete clearing or white hemolysis. 2. Alpha hemolysis, which is incomplete clearing or green hemolysis. 3. Gamma hemolysis, which is no hemolysis. In this picture, you can see different types of hemolysis on blood agar. Second type of special media is enrichment media. Enrichment media is a liquid media used to isolate pathogens from a mixed culture. This media is incorporated with inhibitory substances to suppress the unwanted organism. Examples of enrichment media are selenite F broth for the isolation of Salmonella and Shigella, alkaline peptone water for Vibrio cholerae. Third type of special media is selective media. In selective media, the inhibitory substance is added to a solid media. Examples of selective media are McConkey's medium for gram-negative bacteria, TCBS or thiosulfate citrate bile salt sucrose for V. cholerae, LJ or Lowenstein-Jensen medium for Mycobacterium tuberculosis, potassium tellerite medium for diphtheria bacilli. You can see some selective medias in these slides. Fourth type of special media is indicator media. These media contain an indicator which changes its color when a bacterium grows in them. Example of indicator media are 1. Blood agar, to observe hemolysis and differentiate Streptococcus species. 2. McConkey's medium, to differentiate gram-negative bacteria. 3. Christensen's urease medium, to identify different Enterobacteriaceae species such as Proteus, Klebsiella, and Yersinia. Here is an indicator media. Urease medium. You can see the color change here. Fifth type of special media is differential media. A media which has substances incorporated in it, enabling it to distinguish between bacteria, is called differential media. Example of differential media is McConkey's medium, which can distinguish between lactose fermenters bacteria and non-lactose fermenters bacteria. As you can see in this slide, lactose fermenters bacteria having pink colonies, while non-lactose fermenters have colorless colonies. Sixth type of special media is transport media. This media is used for transporting the samples. Because delicate organisms may not survive in the time taken for transporting the specimen without a transport media. Transport media examples are 1. Stewart's medium, which is non-nutrient soft agar gel containing a reducing agent. 2. Buffered glycerol saline, which is used for enteric bacilli. If you have any question, you can ask in comment section. If you want a lecture on any topic, you can do a comment. We will make it easy for you.
Thank you for watching. Do subscribe, like, comment, and share.